buried for centuries. One breath pattern. The Nobel Prize just unlocked why it reverses brain decline. And in the next 12 minutes, you will know exactly how to use it. This technique has a name, an origin, a precise biological mechanism. It was documented in ancient Vedic texts and practiced by yogic lineages specifically to preserve cognitive function into old age. It is not a meditation. It is not relaxation. It is a targeted cellular intervention, and the science finally caught up. Brain decline is not a disease. It is oxygen starvation at the cellular level. The neurons in your prefrontal cortex, the tissue responsible for memory retrieval, executive function, and processing speed, are mitochondria dense. They are the highest oxygen-consuming cells in the human body, and after the age of 40, the efficiency of that oxygen delivery begins to degrade. This is not opinion. This is metabolic physiology. The mechanism is a molecule called HIF-1 alpha, hypoxia-inducible factor 1 alpha. It is the master regulator of your body's cellular oxygen response. When oxygen availability drops briefly and then recovers, HIF-1 alpha activates. It triggers the expression of genes that repair blood vessels, stimulate the growth of new neuromitochondria, and upregulate neuroprotective proteins. A 2016 meta-analysis in Frontiers in Physiology confirmed that controlled brief oxygen fluctuation activates this cascade in neural tissue. Mainstream medicine addresses the symptoms of this decline. Pharmaceuticals, supplements, cognitive training protocols, none of them touch the root. The root is cellular oxygen dynamics, and the Vedic tradition identified this mechanism, without the molecular vocabulary, 25 centuries ago. What they documented, and why that knowledge was quietly set aside, is the part no one talks about. The Hatha Yoga Pradipika, compiled in the 15th century from traditions dating back over 2,500 years, dedicates an entire chapter to Kumbhaka, the practice of breath retention. The text is precise. It specifies ratios, durations, physiological markers the practitioner should observe. It describes the technique as a method for preserving the fire of the mind into advanced age. This is not metaphor. When you map the described physiological markers onto modern neurological vocabulary, the correspondence is exact. The inner heat they describe is mitochondrial activity. The clarity of the energy channels maps directly to cerebral vascular tone and glymphatic flow. The Vedic physicians understood the mechanism. They simply did not have the imaging technology to name it. So why does the average person in 2025 not know this technique? The answer is not dramatic. It is economic. A breath cannot be patented. A pranayama protocol generates no licensing revenue, no recurring prescription, no proprietary formulation. The clinical research infrastructure in neurology is funded, in large part, by pharmaceutical development pipelines. Research that leads to a patentable molecule gets funded. Research that validates a breath pattern does not attract the same capital. This is not suppression, it is selection. The knowledge was never destroyed. It sat in Sanskrit libraries, in yogic lineages, in Ayurvedic medical traditions, accessible, documented, simply unprofitable to amplify. Until 2019. Because in 2019, 3 scientists won the Nobel Prize, and the mechanism behind Kumbhaka became impossible to ignore. The 2019 Nobel Prize in Physiology or Medicine was awarded to Dr. Greg Semenza, Dr. William Kaelin Jr., and Sir Peter Ratcliffe. Their discovery, the molecular mechanism by which cells sense and respond to oxygen levels. Specifically, they identified HIF-1 alpha, hypoxia-inducible factor 1 alpha, as the master switch that activates when cellular oxygen drops. When oxygen is low, HIF-1 alpha accumulates. It binds to DNA. It switches on a cascade of gene expression, erythropoietin production, VEGF for new blood vessel growth, and critically, in neural tissue, the upregulation of BDNF, brain-derived neurotrophic factor. BDNF is, by current neuroscience consensus, the primary molecule responsible for neuroplasticity. It drives the formation of new synaptic connections, protects existing neurons from apoptosis, and is directly correlated with memory consolidation in the hippocampus. Here is where the ancient text and the Nobel science converge. Kumbhaka, breath retention, creates a controlled, brief, repeatable drop in blood oxygen saturation. Not dangerous hypoxia, not deprivation. A precise, voluntary, recoverable dip, what researchers now call intermittent normobaric hypoxia, the kind that activates HIF-1 alpha without triggering cellular stress responses. A 2020 study published in the Journal of Physiology demonstrated that short, controlled apneic intervals produce measurable HIF-1 alpha accumulation in peripheral blood, the same systemic signal that reaches neural vasculature. A 2018 study in Frontiers in Human Neuroscience used fMRI to confirm that controlled Kumbhaka practice alters cerebral blood flow patterns in regions associated with executive function and memory retrieval. The ancient practitioners did not know what HIF-1 alpha was, but they had, through thousands of years of systematic self-experimentation, landed on the exact breath ratio that activates it. The Nobel Prize did not discover something new. It named something ancient. And the technique itself, the exact ratios, the exact duration, what happens inside your brain during each phase, that is what the next section delivers. The technique is called Kumbhaka, specifically Antara Kumbhaka, internal retention, the pause after the inhale. This is the protocol. Inhale through the nose for a count of 4. Slow, controlled. Filling the lower lungs first, then the middle, then the upper chest. This is not a deep breath in the casual sense. It is a structured, sequential inflation that maximizes diaphragmatic engagement and activates the parasympathetic branch of the autonomic nervous system. Then hold. Full lungs, glottis sealed. Count of 16. During this retention phase, several things happen simultaneously. Intrathoracic pressure increases, momentarily reducing venous return to the heart and creating a brief, controlled drop in arterial oxygen saturation. Carbon dioxide accumulates in the blood. The Bohr effect intensifies. CO2 buildup causes hemoglobin to release oxygen more readily to tissues, including neural tissue. And critically, HIF-1 alpha begins to accumulate in cells responding to the oxygen signal. This 16-count window is the activation window. Then exhale through the nose for a count of 8. Slow, complete. This restoration phase is as important as the retention. The return to normoxia after the controlled dip is what triggers the full HIF-1 alpha response cascade. The gene expression, the BDNF release, the vascular repair signals. The ratio is 4 to 16 to 8. Inhale, retain, exhale. Begin with 3 cycles per session. First week only. The practitioner will notice in the first session a mild warmth behind the eyes, a sharp clarity in the minutes following, and in some cases a subtle pulsing sensation in the temples. This is increased cerebral perfusion pressure normalizing after the retention phase. After 21 days of consistent daily practice, documented changes include measurable increases in cerebral blood flow velocity, normalized nitric oxide bioavailability in vascular endothelium, and, in studies using cognitive performance metrics, statistically significant improvements in working memory scores. Practice in the morning, before food, before screens. The neurological environment at that hour is optimized for this intervention. 30 days of consistent practice produces measurable, specific outcomes. BDNF expression in the hippocampus increases. The molecular substrate of memory consolidation becomes more active. The glymphatic system, which clears metabolic waste from neural tissue during sleep, operates with greater efficiency when cerebral blood flow patterns are normalized. And Kumbhaka directly restores those patterns. Nitric oxide production in the vascular endothelium recovers, improving cerebral autoregulation, the brain's ability to maintain stable blood flow under metabolic demand. These are not subjective improvements. These are measurable biological outcomes, observable in blood work, in imaging, in validated cognitive assessments. This technique was available 2,500 years ago. The Vedic physicians who developed it were not mystics chasing transcendence. They were physiologists working without laboratories, deriving protocols from systematic observation of the human body. The Nobel Prize did not validate ancient wisdom. It confirmed that ancient wisdom had already been correct. The breath was never lost. It was buried. Now, it is yours.